Introduction. Background of study. Frog play vital ecological roles as predator, prey, and bioindicator due to their sensitivity to environmental change. Their dual life stage contribute to nutrient cycling and energy transfer. However, global amphibian populations are declining from threats such as habitat destruction, climate change, and invasive species. Southeast Asia, including Malaysia, is a biodiversity hotspot for amphibians. Yet, many regions remain understudied. LATA KEDING in JELI, Kelantan, with its diverse habitats, is an ideal site for amphibians, but little is known about its frog diversity. Documenting frog species in LATA KEDING is essential for understanding their distribution, ecological roles, and traits, providing critical data for conservation and the protection of Malaysia's amphibian population.
Next, we go to the problem statement. LATA KEDING JELI Kelantan is an area of tropical forest which has a potentially good variety of frog species. However, scientific information about frog species is still very minimal in this area, hence making the effort of biodiversity conservation quite challenging. Besides, threats such as development and habitat degradation can affect frog population. Therefore, studies are needed to identify species diversity and support sustainable conservation strategies. Next, the objective of this research is to identify the diversity of frog species in LATA KEDING JELI Kelantan. Next, we go to scope of study. This research was carried out in LATA KEDING JELI Kelantan, which included tropical forest habitats and watery areas like river. It focused on the gathering of data on the diversity of frog species through direct observation and morphological analysis. It also checked environmental factors like humidity, temperature, and habitat structure to understand the species distribution. This study has been carried out for a month to obtain complete data.
Significance of study. This study was important to understand the biodiversity and ecosystem health in LATA KEDING JELI Kelantan. Frogs were bioindicators that reflected the impact of environmental change like habitat loss and climate change. The research also helped in conservation effort by providing data on frog species diversity and their role in maintaining the ecosystem balance. As better outcome, this study produced a list of frog species in LATA KEDING, possibly discovering new or unique species. It also provided insight into their population size, distribution, and habitat preference. The findings identified endangered species, supported conservation efforts, and demonstrated the overall health of the ecosystem using frog as indicator.
Next, I will present the literature review. First, frog species potentially present in LATA KEDING. 3 amphibian species from 3 families: the Asian common Latiff, Melanostitis, the Asian grass frog Vijaya limnocaris, and the 4-line tree frog Polypedids leucomistax were identified in a preliminary survey carried out by Latiff 2020 in the Delta Tumpak Mangrove Forest, Kelantan. Commonly found in a variety of locations throughout Malaysia, these species may occupy comparable ecological niches in LATA KEDING. Next, we move to second amphibian research in Kelantan. Compared to other peninsular Malaysian states, there is very less research on the variety of frog species in Kelantan. A May 2022 study on LATA KEDING's fauna richness did not include amphibian surveys, but it did emphasize the ecological significance of the area's freshwater system. However, a foundation for comprehending the possible variety of frog in LATA KEDING is provided by the result of analogous hermetofauna research conducted in neighboring localities. In their investigation of the Tasik Bara Ramsar sites, Hermetofauna Norhayati 2005 identified species such the Malayan pond frog, Rana erythrae, and the 4-line tree frog, Polypedid leucomistax. These species are probably LATA KEDING candidate because of their comparable habitat characteristic. Likewise, the region could possibly be home to the Malaysian painted frog, Calola pastra, which is renowned for its ability to adapt to wetland environment. Third, research gaps in amphibian studies in Kelantan. The unequal distribution of amphibian studies in Peninsular Malaysia, with some states like Kelantan being underrepresented, was brought to light by research conducted by Chan and Ahmad 2022. This highlights the necessity for focused hermetofauna surveys in places like LATA KEDING to improve our knowledge of amphibian diversity in Malaysia and points to a substantial research gap. Next, the last one is rare and endemic amphibians of Kelantan. In Gunung Baso, another forest reserve in JELI, Kelantan, Nursyahirah Muhammad, 2022, investigate amphibian species. Among the uncommon and endemic species they discovered were Limnonetus laticeps and Ansonia penangensis. Given the similarities in habitat between LATA KEDING and Gunung Baso, it is possible that these species are also found there.
Okay, next we move to the materials and method. So we go to the study area, which is the LATA KEDING JELI Kelantan is located in the northern region of peninsular Malaysia within a tropical rainforest ecosystem with desert habitats such as freshwater streams and rainforests. This area is suitable for amphibians, particularly frogs. The study cover forest region near water bodies, making it a potential site for amphibia biodiversity research. So we move to the material, which is we will use the sampling equipment, which is hand net, pitfall trap, measuring tape. For field observation tool, we will use the binocular, headlamps, and digital camera. And for environmental data collection, we will use the thermometer, hygrometer, GPS device, and notebook. And lastly, for morphological study equipment, we will use the vernier calipers plastic bag for temporary frog storage. And next, we move to the data collection. We have 3 data collection we will use, which is visual encounter service. We will conduct transit walk for 2 hours per season in the morning and the night in area near rivers and forests to identify and capture frogs. And next, we will pitfall trap placed on the forest floor near water bodies to capture frog, check regularly. And lastly, the data collection, we will use environmental data collection, which is temperature, humidity, and habitat characteristic were recorded at each observation sites. Next, for data analysis, we have 3 data analysis. First, species richness and diversity index. We will use the Shannon-Weines and Simpson index to calculate and to assess species richness and distribution. Second, environmental correlations analysis. The relationship between temperature, humidity, and habitat structure with species distribution was analyzed. And lastly, spatial mapping. GPS data were used to map frog distribution and identify biodiversity hotspots.